In the previous class, we were discussing about the properties of cloning vectors. The first property is the cloning vector should possess an ORI. So this is the gene from where the DNA replication begins. And the cloning vector with an ORI which supports high copy number should be selected. The second important property is cloning vector should possess a selectable marker. Now, selectable marker helps whether the transformation has occurred or not, whether the desired gene has entered into the host or not. Now, generally, we use antibiotic resistance genes as selectable markers. The third important property of a cloning vector is the cloning vector should possess single cloning site or single recognition site for one type of a restriction enzyme. Now that means what? For example, imagine this is, I have chosen plasmid as a vector. Now, this plasmid is having GAA-TTC, GAA-TTC, you know that. So this is the recognition sequence for which enzyme? EcoR1. So it is a recognition sequence for EcoR1. So for one restriction endonuclease enzyme, there should be single recognition sequence on the cloning vector. Now, we are using this enzyme and cutting this vector. So EcoR1 cuts between G and A, which you know it. So EcoR1 recognition sequence is GAA-TTC from 5' to 3' on both the strands. And there should be one recognition site or one cloning site for one restriction endonuclease. Now question is, what happens if there are two restriction sites? So imagine if there is one restriction site, EcoR1 is cutting here and the plasmid will be breaking at this point. But imagine if there are two restriction sites for these enzymes. Imagine somewhere here also GAA-TTC is present. Now in that case, what happens if we use this EcoR1 enzyme? Here also it will break. Now that means what? The plasmid will be cut upside and even in the down region. So that means what? Where do we have to insert the desired DNA fragment? So it will become a complex process. Now that is the reason why for one restriction endonuclease, there should be one recognition site. For one restriction endonuclease, there should be one restriction site. For EcoR1, there should be one restriction site. If you are using some other exam like other enzyme like HIND2, there should be one restriction site for HIND2. So that means what? A cloning vector should possess a single cloning site or a single recognition site for one type of restriction enzyme. The third important property is a cloning vector should have low molecular weight. That means the size of the vector should be less. The molecular weight of the cloning vector should be low, such that it will be easy to handle such kind of a vector. And the next important property is the cloning vector should possess a region of protein. So this is a gene on the cloning vector which produces a protein, and these proteins are useful for the replication of the vector. Region of protein. It is the gene sequence in the cloning vector. What is the function of this gene? This gene produces proteins, and which these proteins are involved in the replication of cloning vector. So these are the properties of a cloning vector. It should possess an ORI, and the ORI which supports high copy number is preferred. Second thing, a cloning vector should have a selectable marker. So selectable markers are generally we use these antibiotic resistance genes. And the third important property of a cloning vector is for one restriction enzyme. So we are using restriction enzymes to cut the cloning vectors, right? So for one restriction enzymes, there should be single cloning site or single recognition site. Right. The next important property is it should have a low molecular weight. The cloning vector should have a low molecular weight or less size. And the cloning vector should also have region of protein. So this is a gene sequence on the cloning vector which produces proteins, and these proteins are involved in the replication of the cloning vector. So these are the important properties of the cloning vectors. Now let's go with some of the examples of the cloning vectors. So we have different types of examples like plasmids, cosmids, bacteriophages, yak, back type of plasmids. So all these plasmids we will learn in the next classes. First we will start with the plasmid. So the first example of the cloning vector is plasmids. We know that plasmids are small circular extra chromosomal DNAs present in bacteria. Generally they are present in bacteria, and you may also see them in archaebacteria and some yeast and even some fungus. Even yeast is also fungus. So generally we observe plasmid in prokaryotic cells such as bacteria and archaebacteria. But you can also find them in some eukaryotic cells such as fungal cells, yeast cells and all. Right. Plasmids are having the capacity of replication autonomously. Now what does it mean? It means that the replication of the plasmid is independent of the chromosomal replication. So plasmids have the capacity to replicate autonomously in a cell. And generally we use plasmids as a cloning vectors in rDNA technology. And there are also artificial plasmids which are constructed by various scientists. Now let's check on them. So here goes. The first artificial cloning plasmid which is PBR322. So this is a first artificial and widely used plasmid which is PBR322. So normal plasmid we make some changes and we prepare such artificial plasmids. The example is PBR322. It is a first widely used artificial cloning plasmid. Here P stands for plasmid. So P is here lowercase, small p, P means plasmid. B here means Bolivar and R is Rodriguez. So BR are the scientist names who have constructed this plasmid PBR322. So B stands for Bolivar and R for Rodriguez. These are the scientists who have constructed this plasmid. And 322 is the identification number of this specific plasmid. So for every plasmid which they are constructing in the laboratory, they give names. So what is the number given for this plasmid is PBR322. 322 is the identification number of the plasmid. So similarly, other type of plasmids have also been constructed like PBR325, 327, 328. So these are also artificial plasmids. And along with that, you can also observe some artificial plasmids, PUC19. So here UC stands for University of California. So this plasmid is constructed by the students of the scientists of the University of the California, PUC19. And here PUC101. It is also constructed by the scientists of the University of California, and 101 is the identification